Welcome to this video. In this video, we are talking about the nucleus. Nucleus is present inside the eukaryotic cells. It is a membrane-bound organelle containing chromosomes and nucleolus. It controls all the cellular activities. So, it is referred to as the controlling center of the cell. Nucleus is the largest organelle of the cell and it's present in all eukaryotic cells. It functions as the heart of the cell. The nucleus acts as the brain of the cell. However, it is absent in RBC of man and conified cells in the skin, hair and nail. History of nucleus. Nucleus was first discovered by Robert Brown in the year of 1831. The term nucleus was derived from the Latin word kernel or nut. Properties of nucleus. Number. Generally, a cell contains only one nucleus, but sometimes two or more nuclei are present. Based on number of nucleus, eukaryotic cells are classified into following types are anucleate cell, mononucleate cell, binucleate cell and multinucleate cell. Anucleate cell. In anucleate cell, the nucleus is absent. Example, RBC of man. Mononucleate cell. In mononucleate cell, single nucleus is present. Example, amoeba. Binucleate cell. In binucleate cell, two nuclei are present. Example, paramecium. Multinucleate cell. It contains many nuclei. Example, opalina. Position. The position of nucleus in a cell varies. Usually, it is situated in the center of the cell. Shape. The shape of the nucleus varies in most of the eukaryotic cells. For example, RBC is the example for spherical shaped cells in nucleus. Spirogyra is the example for cylindrical shaped cells. Size. Size of the nucleus is not constant. It varies from 11 to 25 μm. For example, granulocytes. Chemistry. Nucleic acids are the most important constituent of the nucleus. Nucleic acids are of two types, namely deoxyribonucleic acid and ribonucleic acid. Lipids occur in the form of lipoprotein and phospholipids. It comprises about 3.1% of the total weight of the nucleus. A few important enzymes present in the nucleus are nucleoside phosphorylase, ribonuclease, etc. The inorganic compounds usually found in the nucleus are salts of calcium, iron and zinc. Structure of nucleus. Different components of the nucleus are nuclear membrane, nucleoplasm, chromatin, chromosomes, nucleolus and nuclear pore. Nuclear membrane. Every nucleus is surrounded by a double layer membrane called the nuclear membrane or nuclear envelope. This membrane separates the nucleoplasm from the cytoplasm. It is made up of two membranes, an outer membrane and an inner membrane. Each membrane is composed of phospholipids arranged in a bilayer. The inner and outer membrane are separated by a perinuclear space, which is about 150 to 300 Armstrong. Outer nuclear membrane continues with rough endoplasmic reticulum. Both the rough endoplasmic reticulum and outer nuclear membrane are covered with ribosomes. Inner nuclear membrane is attached with nuclear lamina. Nucleoplasm. The term nucleoplasm was coined by Strasburger in 1882. The nucleus is filled with a gelatinous sticky fluid known as the nucleoplasm or nucleus sap. The chromatin threads remain suspended in the nucleoplasm. In addition, there may be larger bodies which stain like chromatin threads, and hence they are known as chromatin nucleoli or false nucleoli. The nucleus sap contains organic and inorganic substances like nucleic acids, proteins, enzymes, and minerals. Chromatin. The term chromatin was coined by Fleming in 1879. Chromatin are classified as euchromatin and heterochromatin. Euchromatin. The light staining and less condensed regions of chromatin is termed as euchromatin. Heterochromatin. The darkly stained and highly condensed portion of chromatin is termed as heterochromatin. There are lightly stained thread-like bodies embedded in the nucleoplasm called the chromonemata, which form a network called the chromatin reticulum. Chromosomes. The chromatin network readily stains with basic dyes. They represent chromosomes. In Greek, chromo means color and soma means body, due to their property of being very strongly stained by particular dyes. Centromere. The constricted region of linear chromosomes is known as the centromere, although these constrictions is called the centromere. It is usually not located exactly in the center of the chromosomes, and in some cases which is located almost at the chromosome's end. The region on either side of the centromere are referred to as the chromosome's arm. The centromeres serve both as the sites of association of the sister chromatids and as the attachment sites for microtubules of the mitotic spindle. Origin of replication. The origin of replication is a particular sequence in a chromosome at which replication is initiated. One chromosome contains multiple origin of replication. Chromosome number. All eukaryotic cells have multiple linear chromosomes. Chromosome number in eukaryotes are species specific. Every species maintains a characteristic number of chromosomes. Depending on the eukaryotic organism, the number of chromosomes vary from 2 to several hundreds. Eukaryote can be monoploid having one set of chromosomes, diploid having two sets of chromosomes, and polyploid having more than two sets of chromosomes. The majority of eukaryotes are diploid. Chromosome number in different eukaryotic organisms. The species Saccharomyces cerevisiae in budding yeast having 16 number of haploid chromosomes. The species Schizosaccharomyces bombi in fission yeast having 3 number of haploid chromosomes. The species Enorhabditis elegans having 6 number of haploid chromosomes. The species Arabidopsis thaliana having 5 number of haploid chromosomes. The species Drosophila melanogaster having 4 number of haploid chromosomes. The species Tetrahymena thermophile having 5 number of haploid chromosomes. The species Homo sapiens in human having 23 number of haploid chromosomes. Nucleus. Nucleus was discovered by Fontana in 1781. The nucleus is the larger structure in the nucleus. Nucleoli are made of proteins, DNA and RNA. Each nucleus produced by a nucleolar organizing region. NOR of a chromosome which is termed as nucleolar organizing chromosome. All eukaryotic cells contain at least one such chromosomes. Its number may be 1 or more, but 1 to 4 being the most common. The number of nucleoli per nucleus also differs. The yeast cell contains one relatively large nucleus with respect to its nuclear value. At the other extreme, xenophous oocytes contain over 1000 nucleoli per nucleus. It is a site of transcription of ribosomal RNA and assembly of ribosome. It is responsible for synthesizing large nascent precursor ribosomal RNA, that is 45S in mammals, and the processing of this RNA to yield mature ribosomal RNA of 5.8S, 18S and 28S. The concomitant assembly of RNAs with incoming ribosomal proteins generate small and large ribosomal subunits that then pass into the cytoplasm. Nucleolus structure. It consists of three major regions. Fibrillar centers. It containing rRNA genes in the form of partly condensed chromatin. Fibrillar component. It surrounds the fibrillar centers, which contains RNA molecules in the process of transcription. Granular regions. It is the outermost region having mature ribosomal precursor particles. Nucleolar organizing region. The nucleolus organizing region or nucleolar organizer is a region of chromosomes around which the nucleolus forms. In the human, the 5 chromosomes contain nucleolar organizing regions, which can be identified as secondary constructions on metaphase chromosomes. However, not all secondary constructions are nucleolar organizing region. The position of secondary construction in a chromosome is specific and species specific. As compared to secondary constructions, the primary constructions are also referred to as centromere that attaches sister chromatids together. The difference between these two constructions can be noticed during anaphase, as chromosome can bend only at the site of primary constructions. The area of a chromosome distal to nucleolar organizer is called satellite or trabant. A chromosome with a secondary construction usually associated with a nucleolus, which is called as sat chromosomes. Nuclear pore. The nuclear membrane contains many pores called nuclear pores. The pores regulate the passage of molecules between the nucleus and cytoplasm. There is a ring-like structure called annulus, made up of vertical fibers. Diameter of nuclear pore is 800 angstrom. There is a lumen part which is filled with nucleoplasmid through the substances are transferred. The space which is allowed for entry of substance is 500 angstrom. Shape of nuclear pore is octagonal. Number of pores varies from 1000 to 10,000 per nucleus. Each nuclear pore consists of a large multiprotein complex and termed as nuclear pore complex. The inner and outer membrane fuses at the nuclear pore complexes. Nuclear pore complexes consist of multiple copies of roughly 30 different proteins known as nucleoporins. The nuclear matrix is a network of protein filaments in the nucleus which provides a structural framework for organizing chromatin while facilitating transcription and replication. The term nuclear matrix is defined as the biochemical entity that can be isolated from cell's nuclei after sequential extraction with non-ionic detergents, nucleases, and high salt buffers. The ultrastructure of the nuclear matrix consists of a nuclear lamina and a network-like internal nuclear matrix. The structural portions of the nuclear lamina have been well characterized, but the structural biochemistry of the internal nuclear matrix is less known. The nuclear lamina is a network of intermediate filaments present on the nuclear side of the inner membrane. It is made up of lamin proteins. The nuclear lamina provides the mechanical support to the nucleus. It links the internal nuclear matrix to the nuclear membranes. Transport through nuclear pores. Bidirectional transport of molecules occurs through nuclear pore between the cytosol and the nucleus. RNA molecules and ribosomal subunits are exported to the cytosol, while all of the proteins that function in the nucleus are synthesized in the cytosol and are then imported. The molecules are actively transported in to and out of the nucleus. Proteins imported to or exported from the nucleus through nuclear pores contain a specific amino acid sequence that functions as a nuclear localization signal or a nuclear export signal. Several types of nuclear localization and export signals have been identified. Each type of nuclear transport signal interacts with specific receptor protein described as export in or import in. RAN shuttles across the nuclear envelope through nuclear pores but is concentrated in the nucleus because of nucleus transport factor 2 mediated active import. In the nucleus, a high concentration of RAN-GTP is generated by nucleotide exchange. This is catalyzed by chromatin bond guanine nucleotide exchange factor. RAN-GTP causes the dissociation of imported complexes which contains proteins that carry a nuclear localization signal by binding to import in and ejecting the cargo. RAN-GTP also promote the assembly to export complexes containing protein with a nuclear export signal. In the cytoplasm, RAN-GTP meets GAP which stimulates GTP hydrolysis at the export complexes dissociate. Functions of nucleus. Nucleus controls majority of the activities of cells. It is a regulatory organelle in cell metabolism. The nucleus contains DNA molecules in its chromosomes. It plays a significant role in heredity. First and foremost, it is possible to duplicate one's DNA in the nucleus. This process has been named DNA replication and creates an identical copy of the DNA. Secondly, the nucleus is the part of transcription. Transcription is the process of creating different types of RNA from DNA. Transcription would be a lot like making individual copies of the human body's instruction that can then be passed out and read by the rest of the cell. The central rule of biology states that DNA is copied into RNA which is turned into protein called translation.